Hey everyone! Hope you're all doing alright today. Today in O-Level Biology, Lesson 8, Transport in Plants. Large plants need transport because roots obtain water and minerals while leaves make sugars. Root hair cells have long projections, giving a large area in contact with soil water. Each branch asks a different question about life, plants, animals, microbes, inheritance, or interactions. The living world is one system studied from several angles. Water enters from higher soil water potential by ounce MOH through partially permeable membranes. Mineral ions may enter by diffusion or by active transport when their concentration is lower in soil than in root cells. Water crosses the cortex and reaches Xylem ions lower cell water potential and can help maintain water uptake. Xylem vessels form long hollow tubes from dead cells whose end walls are lost. Lignin waterproofs and strengthens their walls, preventing collapse under tension and supporting the plant. Each branch asks a different question about life, plants, animals, microbes, inheritance, or interactions. The living world is one system studied from several angles. Xylem carries water and dissolved mineral ions mainly upward. Water supplies photosynthesis, maintains turgor and transports minerals. Read left to right, inputs enter, the living process transforms them, outputs leave. Name what goes in and what comes out before memorizing the word equation. Phloem consists of living sieve tube elements joined through sieve plates and supported by companion cells rich in mitochondria. It translocates sucrose and amino acids from sources to sinks, potentially in opposite directions in different tubes. Transpiration is loss of water vapor from aerial plant parts, mainly through stomata. Water evaporates from moist mesophyll walls into leaf airspaces and diffuses out down a water vapor gradient. Each branch asks a different question about life, plants, animals, microbes, inheritance, or interactions. The living world is one system studied from several angles. This loss lowers leaf water potential and creates tension in Xylem. Cohesion between water molecules maintains a continuous column pulled upward. Adhesion helps water remain against vessel walls. This transpiration stream links evaporation at leaves to uptake at roots, without a mechanical pump like a heart. Light usually opens stomata and warms leaves, increasing transpiration. Higher temperature speeds evaporation and diffusion. Wind removes humid air around a leaf, steepening the gradient, whereas high humidity reduces it. Low soil water can cause guard cells to lose turgor and stomata to close, conserving water but restricting carbon dioxide uptake and photosynthesis. Read left to right, inputs enter, the living process transforms them, outputs leave. Name what goes in and what comes out before memorizing the word equation. Xerophyte features such as rolled leaves, hairs, thick cuticles, and sunken stomata trap humid air or reduce exposed area. A bubble potometer estimates water uptake, not transpiration directly, because some absorbed water supports photosynthesis, growth and turgor. Read left to right, inputs enter, the living process transforms them, outputs leave. Name what goes in and what comes out before memorizing the word equation. Seal joints, cut a chute underwater, introduce one bubble and measure distance per time. Bore area converts distance to volume. Change one factor, allow equilibration, and repeat with comparable leafy shoots. A faster bubble does not prove greater water loss unless leaks and leaf area are controlled. Ringing experiments can show phloem transport when sugars accumulate above removed bark. Potometer volume rate equals PIR squared x bubble distance time. Compare rates per unit leaf area if shoots differ, and identify uptake as the measured dependent variable. In a tube of area 0.50 mm2, a bubble moves 80 mm in 200 s. Uptake rate is 0.50 x 80, all over 200 equals 0.20 mm3s. Do not say water is actively transported up Xylem, that all STOH mutu close in light, or that FLOHM carries only downward. State how each environmental factor changes a gradient. Practice. Explain why wind increases transpiration. Answer. Wind removes humid air near STOH mutu, lowering external water vapor concentration and maintaining a steeper dFY-ozon gradient from leaf to air. Watch the numbers land in the matching symbols. Check that units cancel cleanly. If they do not, the setup is wrong even before the arithmetic. Practice. Tube area is 0.40 mm2, a bubble travels 150 mm in 300 s, calculate uptake rate. Answer, 0.40 x 150, all over 300 equals 0.20 mm3s. Watch the numbers land in the matching symbols. Check that units cancel cleanly. If they do not, the setup is wrong even before the arithmetic. Okay, that's it for transport in plants today. Next time in O-level biology we'll get into transport in humans. Stay curious and see you in the next lesson.